Hello everyone. My name is Mahfuz Rahman. I am an associate professor and plant pathology extension specialist at West Virginia University in Morgantown. Before moving to WVU in 2011, I worked at NC State on strawberry integrated disease management. After that, strawberry was not my primary research focus. But lately, I started working on a few projects on strawberry disease management. So, together with epidemiology and management of strawberry anthracnose, my talk today will include some results from those studies. As strawberry growers, you know that disease is always a big concern because all parts of strawberry plants, including root, crown, petiole, runner, and fruit, can be affected with many different types of diseases, from which anthracnose is a major concern. So what you are seeing here, some of the photos of diseases at the left-hand side, upper left corner. Here, the black root rot complex that is caused by many different types of fungi, including Rhizoctonia, Pythium, and also leash and nematodes play a role in black root rot complex. So when these organisms affect the root system loses all the feeder roots and it turns kind of black. What you are seeing here is kind of red tail appearance compared to the healthy one. As a result, plants get stunted and uh, yield seriously reduced. So black root rot complex is primarily a problem when small growers or organic growers cannot use synthetic fumigants or any kind of fumigation, or growers grow strawberries for a few years in the same plot without any option for rotation, such as like what many growers are still in the Northeast and in the Mid-Atlantic doing so-called perennial matted row system. So here you can see another photo is showing a plant is collapsed, that's next to the healthy plant. So plant collapse mainly can happen due to crown rot. It can be caused by Phytophthora or anthracnose crown rot, Coleotrichum gliosporoides. So you have the option to actually dig out that plant with the root system and take a longitudinal cut of the crown to see the what kind of discoloration you are seeing. If there is a discoloration that is orange to reddish brown and then marbly appearance, that will be an indication of anthracnose crown rot. And at the lower left side, what you are seeing is anthracnose fruit rot. I think many of you are familiar with this disease because this is the most concerning disease of strawberry. Sunken black lesions, but these strawberries with lesions, if you keep it under high humidity, they will produce tons of spores that will turn these lesions kind of peach or salmon colored. So the problem with anthracnose is that under low level of infection, these infections remain latent or asymptomatic. However, when the disease pressure is very high, you can see lesions or symptoms on petioles, on runners, and even on the leaf blades, what you are seeing here with some black irregular lesions. These are caused by Polytroticum gliosporoides. And also here you can see the sunken areas on the petiole. And these petioles, infected, suspected petioles or runners, if you put them inside a Ziploc plastic bag for a few days with a moist paper towel, they will sporulate, or you will see lots of sporulation that will be turn the area kind of salmon colored or peach colored. That will be an indication that these plants are infected with anthracnose. As strawberry growers, lately what has been happening with this pandemic. And so in case of plant diseases, we call it epidemic. And we need to have a real good understanding what epidemic or epidemiology is. In terms of strawberry plant supply, what we know that plants are grown in the nursery, and then how these plants are moving to the flax production facilities, and from there it moves to the fruiting field. So we really need to have a good understanding how these plants are produced and at what stage they can get infected. Even after infection, why these plants keep moving from one production system to another. The short answer is the infection is taking place, but we do not see the symptom. Latent infection or asymptomatic infection. We call them also hidden enemies. The symptom can appear only when the infection level is very high or plant is in a different maturity, like when the fruits start ripening. So what do we know about plant supply? What do we know about the infection? And what are the best options for us to minimize the impact of these diseases? Obviously, our strawberry nursery growers start with healthy, disease-free tissue cultured plants, but they have to multiply those plants in the nursery production system in many cycles. That's why the risk is coming from the infection can come from the surrounding areas. These two groups of anthracnose causing fungi, Coleotrichum equitatum group and Coleotrichum gliosporoides group, both of them have very wide host range, especially Coleotrichum equitatum. They can cause disease on many different types of vegetables and fruits, very similar to Coleotrichum equitatum. Gliosporoides can also can be on many different alternative hosts. So when nursery growers are growing plants with the rain splash, those plants can get the initial inoculum. And those inoculum keep multiplying on plants, but still, if not very high level of infection, they will not produce any kind of symptom. So when those tips or even bare root plants are coming directly to your fruiting field, or tips are going to the flag production stage, those flag production obviously that includes very high mist application. So this infection can further spread. But as the plug production stage is relatively short, only 4 to 6 weeks, the level of infection staying more or less the same, or it can slightly multiply at that stage, but from the surrounding area coming from the surrounding area is relatively low. So what I have been working before and also now asking the questions, how can we know where the infection is coming from and what level of infection we have on the plants as latent? And if we can make that information available to different levels, nursery growers or flag producers or to the fruit producers, can that help minimizing the impact of the disease? So we are now working on a share-funded project to diagnose these latent infections on tips and then get that information available to flag producers so flag producers can decide what to do with those infected plants, potentially infected plants, and that those plants should be sold to the fruit producers or those need to be treated at what stage. So in any situation, what you are seeing, the epidemiology of anthracnose, these two different groups, they have similarities and dissimilarities in their life cycle. What we know that crown rot group or Coleotrichum gliosporoides group is a high temperature loving organism. So uh, at flag production or even in the nursery production stage in eastern North Carolina, we have seen Coleotrichum gliosporoides infection is higher compared to the western North Carolina on the mountain. But even on the mountain, those plants can get infected with Coleotrichum acutatum. And both of these can be rain splashed from infected to healthy plants and even from the surrounding areas. So fungicide application is obvious to keep these plants healthy. And we know those different fungicides are effective against anthracnose, but QOYs or strobilurins are the most effective groups. But at the same time, if we apply these strobilurins fungicides on strawberries for managing anthracnose, fungal population can become resistant against those fungicides. So if we know what products have been used in the nurseries, if any of these products have been used, if any of these products have been used in the flag production stage, if the flag producers used, and that information needs to be taken to the fruit producers. So they know what is the status of their plants. They got, either those plants have any latent infection, and if any products have been used. So fruit producers can take informed decision about their spray schedule. If you see in your fruit production field, this is the symptom. What you see is the blossom blight due to anthracnose, Polytotricum equitatum infection. As you can see also, some of the green fruit are affected here. So if this is the first thing you see in your fruit production field, obviously that is very nerve-wracking. And then at that stage, it is very, very difficult even if you initiate a spray program at that time. So uh, while I have been working at NC State, we did some work to diagnose quiescent or latent infection on strawberry foliage and then trying to make a prediction whether in the fruiting field you will get anthracnose fruit rot or not. We published that in California Strawberry Commission production research report in 2011. At that time, we evaluated plants by taking petioles and leaf blades from three different fields. One field was at the research station in Castle Hayne, North Carolina. And what you are seeing that actually we inoculated those plants before planting. So at the fruiting field, level of infection kept going up, 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 and up. And obviously we got close to 60% latent infection that translated to 37% anthracnose fruit rot, whereas on the other two fields, we got the very low level of latent infection, and we did not get any anthracnose fruit rot, or at the very late stage, only very low level of anthracnose fruit rot. So we asked the question whether we can do the latent infection, and if we find higher percentage of latent infection, whether initiating a spray schedule can reduce the latent infection. Here you can see we did start sampling in December, and then we continued sampling all the way in the spring, and then we applied, when we found that very high level of latent infection, we applied fungicides, and you can see the level of latent infection actually came down. So if you can bring down that latent infection level early in the season, then you have a good sense that during fruit harvest, you may or may not need any fungicide spray, which is obviously a good news for UPIC growers, because you obviously do not want to use any fungicides when your customers are picking strawberries. So as I mentioned, some of those fungicides is an obvious need for keeping strawberry plants disease-free, but they are also very prone to resistance development. So you have to get the information what products have been used previously, and then you need to decide your own spray schedule based on that information. Say, for example, if the previous plug production cycle or in the nurseries, pristine has been used, or any kind of strobilurin have been used, it is probably not a good idea you start with Maribon as an alternate product, or rotation product, because these have the same active ingredients, and the frag group is 7+11. In that situation, the best idea for you will be to bring Captain or Thyram, and Mirabis, has a different active ingredient or chemistries. So now I'll switch gear and then talk a little bit about what we have been doing for managing black root rot complex disease and crown rot. So we hypothesized that if we can colonize the root system of strawberry with beneficial microbes like TerraGrow has 6 different types of beneficial microbes. So we wanted to inoculate the plaque production medium, and then we also did 2 different types of inoculation: pasteurized medium inoculation, and then non-pasteurized or regular planting mix inoculation with TerraGrow. And in one treatment, we kept it non-treated. That is, those plants are taken to the fruiting field, and then in fruiting field, we did some mustard cover crop treatment and anaerobic soil disinfestation treatment. I'll tell you in a second what is being done for anaerobic soil disinfestation. So we ended up having 6 different treatments. Non-treated, that means in the plaque production stage, no treatment, no inoculation of TerraGrow, and those plants, non-treated plants, taken to the mustard cover crop plots. And regular planting mix inoculated with TerraGrow and then taken in the fruiting field, no treatment there. Pasteurized mix treated with TerraGrow and then taken to the fruiting field. And anaerobic soil disinfestation. And also we had one treatment to have the synergistic effect from pasteurized medium that treated with TerraGrow and then planted in the anaerobically soil disinfested plots. So just to give you a snapshot of mustard cover crop that we used for biofumigation, as you can see, we grew the mustard plots in pre-designated, pre-allocated plots, and then 70 to 75% flowering. We macerated the tissue and then incorporated the tissues in the soil. Then we prepared the beds, covered it with plastic mulch. So many growers actually I talked to, they had a mixed impression about mustard cover crop. And also even in our experiment, we did not get very good result. So, but there are few different factors affecting this mustard cover crop. Obviously, we need to use very high glucosinolate containing variety like Caliente 199. And we need to macerate the tissues at the right flowering stage. Macerated tissues needs to be incorporated in the soil right away. And if the soil moisture is not high enough, you need to irrigate. After that, it is a good idea to cover that area with plastic mulch, or you can prepare the bed right away. Anaerobic soil disinfestation, this is very similar idea, adding carbon source in the soil and then incorporating that in the soil, followed by preparing the bed and then saturating the bed by irrigating through drip irrigation. So when you create an anaerobic environment, all the anaerobic microbes flourish in those beds, and they produce like a kind of toxic or suppressive gas, and also many volatile organic compounds that helps in controlling the harmful microbes. So here you are seeing that this work was done in California, that they sprayed carbon source rice bran and then incorporating that in the soil. After that, they prepared the bed with drip irrigation and then covered with plastic mulch and irrigated those beds to saturation. So the results they got actually very encouraging, because in their case, the major problem was verticillium wilt of strawberries. Here you can see the non-treated plot had very high verticillium wilt compared to the ASD plot. What we got from our study, the first thing we actually observed that some plots had lower level of weeds came through the planting holes. What you can see from these lines, from this graph, is that number of weeds per plot was significantly lower in ASD plots, but in other plots there was no reduction of weeds. What is expected from anaerobic soil disinfestation, because there are some fumigation effect coming from ASD. So fruit yield in the second year, you can see the overall fruit production was lower compared to the first year, because we kept those plants for a second harvest. And obviously here you can see that in the second year we got some lower level of fruit harvest, but still significantly higher compared to the non-treated and mustard cover crop. So mustard cover crop did not work in our case. So let's go back and then compare that with the first year yield. In the first year, what we saw only significant yield boost from the pasteurized medium TerraGrow, and then some additive effect or synergistic effect from uh, TerraGrow treatment of the planting medium and also anaerobic soil disinfestation. Here you can see higher level of yield per plant compared to the second year. So you can see after we started with 20 plants in each plot, and then after the first year, obviously, some plants died and most plants died in the non-treated check, also in mustard cover crop treatment. But in the second year, after the end of the second year harvest, we saw more plants died and a significantly lower number of plants survived in the non-treated plot compared to other treatments. So here just to show you the plots at the end of the second year, this plot is non-treated. So basically what you are seeing here, lots of dead plants and also overall plant health pretty much declined compared to mustard cover crop or TerraGrow treated plots and anaerobic soil disinfestation. And this is the plot we actually got the highest number of plant survival and also the plant health very, very good compared to other treatments. What was a synergistic effect of anaerobic soil disinfestation and TerraGrow inoculation of the flag plants. So if you have any questions, you have my email address. Please do not hesitate to send me email. And also for the share-funded project where Dr. Samtani is also a collaborator together with Dr. Alison Smart from University of Maine. So we developed a Qualtrics survey to recruit some beneficiaries for our project. So if you please go ahead and then fill that Qualtrics survey, we will get your information, and then later on, because our project will hopefully will produce more results from our share funded project, and then we can share those results with you over time. Thank you very much.